Hello everyone, welcome back for the next session of Biology Chapter 9, Control and Coordination. Last time we studied about the introductory part. We studied about the nervous system in Hydra, nervous system in Planaria and neural tissues, which includes neurons, that is nerve cells, and neuroglial cells. Today we'll start with synapse. What is synapse? It is a junction between two nerves. You can see here two nerves. This is the first one and this is the second one. And here you can see a gap. It is a junction between two nerve cells with a minute gap. That gap is called synaptic cleft. This gap you can see, it is synaptic cleft, and this allows the transmission of impulse by neurotransmitter. You can see these are all neurotransmitter which helps in passing the message from one neuron to another neuron. Now let us start with properties of nerve fibers. There are several properties of nerve fibers. The first one is excitability or irritability. It is nothing but it is the ability of a nerve to perceive stimulus and enter into a state of activity. That means the nerve fibers becomes active on receiving the stimulus. That is called excitability or irritability. The next one is conductivity. It is the ability to transmit the excitation. Now, the nerve which is excited will transmit this impulse forward. That is called conductivity. Next is stimulus. What is stimulus? It is any kind of detectable change. It may be physical, chemical, or electrical change in the external or internal environment, which brings about excitation in nerves or organs. This is called stimulus. In order to be effective, the stimulus must have a minimum intensity called threshold stimulus. Whenever there is threshold stimulus, the stimulus will be effective. If there is any subliminal, that is weak stimulus, then that weak stimulus will have no effect. But the supraliminal, that is strong stimulus, will produce the same degree of impulse as the threshold stimulus. So, for stimulus, for excitability, it should be strong, that is supraliminal, and not weak, that is subliminal. Next is summation effect. A single subliminal stimulus will have no effect. As I already told you, the subliminal stimulus will show no effect. But if such kind of weak stimuli are given in quick succession again and again, then it may produce an impulse due to addition or summation of stimuli. All or none law. The nerve will either conduct the impulse along its entire length or will not at all conduct the impulse. This is the case in subliminal or weak stimulus. Then refractory period. Refractory period is nothing but it is the time interval during which a nerve fails to respond to a second stimulus, however strong it is, and that time interval is about a millisecond. Next is synaptic delay. The impulse requires about 0.3 to 0.5 milliseconds to cross a synapse. This time is required for the release of neurotransmitter from the axon terminal and excitation in the dendron of the next neuron. That's why it is called synaptic delay. Next one is synaptic fatigue. Now, the nerve impulse which has been transmitted, that transmission across the synapse halts temporarily. This halting is due to exhaustion of neurotransmitter, and this is called synaptic fatigue. Then velocity of transmission. The rate of transmission of impulse is higher in long and thick nerves. It is higher in homeotherms than in poikilotherms. Homeotherms means those who are having constant body temperature, or you can say warm-blooded animals. Poikilotherms, they are called as cold-blooded animals. Now here in homeotherms, the rate of transmission of impulse will be higher than that in cold-blooded animals. The velocity of transmission is higher in voluntary fibers and it is low in involuntary nerves or involuntary fibers. Similarly, it is faster in modulated nerve fibers as the impulse has to jump from one node of Ranvier to the next node of Ranvier. At the synapse where the neurons communicate with one another, the neuron carrying an impulse to the synapse is the presynaptic neuron, and the neuron receiving the input at the synapse is the postsynaptic neuron. A synaptic cleft is present in between the two cells, having a width of about 20 to 30 nanometer between them. The process by which the impulse from presynaptic neuron is conducted to postsynaptic neuron is called synaptic transmission. It is a one-way process carried out by neurotransmitter. Now, types of synapse. There are two types of synapses, that is electrical synapse and chemical synapse. Let us start first with electrical synapse. In this type of synapse, gap between the neighboring neurons is very narrow. The synapse between such close neurons is mechanical. It is found in those places of the body which requires fastest response. For example, defense reflexes. Electrical synapse. In this type of synapse, gap between the neighboring neurons is very narrow. These are certain examples of animals showing electrical impulse. It involves crayfish, coelentrates, annelids, mollusks, arthropods, and some fish. Now here, in the electrical synapse, the electrical connective link is formed between the pre- and postsynaptic neuron. At the gap junction, the two cells are within almost 3.8 nanometer distance of each other. Transmission across the gap is faster, but depends on the connection located at the gap junctions between the two neurons. It is found in those places of the body which requires fastest response. Example, defense reflex. They are bidirectional, allowing transmission in either direction or may be unidirectional. Chemical synapses. These are specialized junctions through which cells of the neural system send chemical signals to the other neurons and to non-neuronal cells such as glands and muscles. Synaptic gap is larger than that of electrical synapse and it is near about 20 to 40 nanometer. A chemical synapse between a motor neuron and a muscle cell is called a neuromuscular junction. There are three components of a typical chemical synapse. That is, the presynaptic terminal, you can say the axonal terminal, the synaptic membrane of the postsynaptic cell, usually on the dendrite of the next neuron or gland cells or muscles, and the postsynaptic neuron. The impulse travels along the axon of the presynaptic neuron to the axon terminal. Most presynaptic neurons have several presynaptic knobs at their ends or terminals. These knobs have arrays of membranous sacs called synaptic vesicles that contains neurotransmitter molecules. Calcium ions are present near the vesicle. When an impulse reaches a synaptic knob, voltage-sensitive calcium channels open and calcium ion diffuses inwards from the extracellular fluid. The increased calcium ion concentration inside the cells initiates a series of events that fuse the synaptic vesicles with the cell membrane of presynaptic neuron, where they release their neurotransmitter by exocytosis into the synaptic cleft. Once the neurotransmitter binds the receptors of the postsynaptic cells, the action is either excitatory or inhibitory depending on the nature of neurotransmitter involved. Now the sodium ion channels opens and the sodium ion diffuse inside the postsynaptic neurons. Now here, if there is a threshold potential, then the impulse travels from one neuron to another neuron. Once the impulse has been transferred across the synapse, now the enzyme like cholinesterase destroys the neurotransmitter and the sodium channel is closed and the synapse is now again ready to receive a new impulse as the degraded neurotransmitter diffuse back into the synaptic knob. Transmission of nerve impulse. The neurons are cells with some special features. The cells can be excited. The nerve impulse is a wave of bioelectrical or electrochemical disturbances passing along a neuron. The transmission of a nerve impulse along the long nerve fiber or axon tube is a result of electrical charges across the neuronal membrane during the conduction of an excitation. Each neuron has a charged cellular membrane with a voltage which is different on the outer and inner side of the membrane. The plasma membrane separates the outer and inner solutions of different chemical compounds but having approximately the same total number of ions. Here you will find sodium ion, potassium ion, chloride ion, some negatively charged protein molecules and nucleic acid are present in the conduction. Tissue fluid has both sodium and potassium, but there is predominance of sodium and chloride while potassium is predominant within the fiber or in the intracellular fluid. This condition is of a resting nerve. It is called polarized state, where excess of sodium is present on the outside, making it positive, and potassium ion is present inside. On the inside there is excess of potassium along with large negatively charged protein molecules and nucleic acid. Some amount of sodium and potassium always leaks across the membrane. The nerve membrane not only has leakage channel but also has many gated channels for sodium and potassium. First we'll talk about leak channel. The sodium-potassium gated channels are separate, so the transport of both these ions is separately done. However, during resting potential, both these gates are closed and the membrane resting potential is maintained. Now, voltage gated channel. This potential difference seen in the resting nerve. Here, the resting potential is approximately minus 70 millivolt because here the difference is between minus 50 to minus 100 millivolt. Now here you will find due to this differential permeability of the resting membrane, which is more permeable to potassium than to sodium ion. This results in slightly more potassium ion diffusing out than sodium ion moving inside and causing slight difference in polarity. The sodium-potassium pump in the membrane actively restores the ions to their appropriate site. Against the concentration and electrochemical gradient, now sodium ion is being forced out and potassium ion is being forced inside the membrane. This process is called sodium pump or sodium-potassium exchange pump. This active process requires ATP energy. The difference in distribution of sodium and potassium on the two sides of the membrane produces a potential difference of minus 50 to minus 100 millivolt, on an average minus 70 millivolt. These channels enable the neuron to change its membrane potential to active potential in response to a stimuli. Here you will find all the positive charge is outside and negative charge is inside. This indicates that it is the resting potential. Generation of nerve impulse. This you can understand clearly through the video. In a polarized state, you will find the extracellular fluid is positively charged whereas the intracellular fluid is negatively charged. During the resting potential, both the gates are closed and the resting potential is maintained. The origin and maintenance of resting potential depends on the original perfect state. Any change or disturbance in the membrane will cause sodium ion to enter into the membrane and lower the potential difference. The voltage gated sodium-potassium channels are special in two ways. That is, they can change the potential difference of the membrane as per the stimulus received, and also the gates operate separately and are self-closing. However, during depolarization, sodium gates open but not the potassium gates. This causes sodium ion to rush into the axon and brings about a depolarization. Now here, due to influx of sodium ion, the ECF becomes electronegative with respect to the inner membrane, which becomes electropositive. The value of action potential is plus 30 millivolt to plus 60 millivolt. This triggers depolarization in the next part. Now the nerve impulse moves from one part to another. One part which becomes depolarized, and the next part will start going to become repolarized. Now here you can see the part site A having extracellular negatively charged and intracellular positively charged, that is, it is depolarized state. Now it will pass the impulse to site B, where influx of sodium ions will take place, and so here again you will find the inside will become positive and the outside will become negative. In this way, here this process of producing a wave of stimulation causing depolarization, repolarization is repeated continuously up to the end of axon terminal. Here it occurs, this change in polarity actually occurs after a short interval called refractory period. Now repolarization. The large number of sodium ion on the inside causes a drop in the permeability of sodium ion, and at the same time making it more permeable to potassium ion by opening the potassium voltage gates and slowly closing the sodium gates. This action is a localized activity. Sodium ion passes out very rapidly as compared to the slow entry of sodium ion. Now here you can see the sodium ion which is present inside along with sodium potassium pump will try to move out and potassium ion will come inside. That means here sodium potassium pumps becomes operational. It is a self-propagating process. This is a modulated nerve fiber having myelin sheath and node of Ranvier. In modulated nerves, the insulating fatty myelin sheath prevents flow of ions between the exoplasm and ECF. The transport pump and gated channels can operate only in the region of nodes of Ranvier, where myelin sheath is absent. The action potential cannot travel as a wave of membrane depolarization. It has to jump from node to node. This process is called saltatory conduction and it is faster. That's all for the day. Thank you. I hope you understood the topic. Let's stop here. We'll continue in the next video. Till then, take care of yourselves.